We start today with Biotechnology Part 1. Biotechnology means all processes of biological modification of different organisms leading to commercial improvement of agriculture, food materials, vaccine production, medicine synthesis, and control of agricultural upgradation. Several terminologies are very much related to the concept of biotechnology. One of the languages, Plasmid. What is plasmid? Plasmid can be defined as a extranuclear piece of free DNA of bacterial origin which serves as a cloning vector or carrier for a native DNA to another cell. So the language goes to cloning vector, which serves as an ideal carrier for DNA for transference. After plasmid, we go with another terminology. The terminology is restriction endonuclease. This group of enzyme is supposed to be for cutting specific segments of DNA. As this can cut the desired segment, so this is called molecular scissor. A very important concept is this molecular scissor, or you may call it chemical scalpel, as recommended by several other biologists. The first identified plasmid is called PBr322. Why the name is PBr? First identified by Bolivar and Rodriguez. Another plasmid is also of very commercial importance, PUC19, but the plasmid that was identified in University of California bearing strain number 19. If we make a schematic diagram of the plasmid PBr322, this drawing is almost like this, where we are getting several sites. This is called ORI. The site represents origin of replication. This is called ROP, which stands as region of replication of plasmid. These are the selectable markers, which is ampicillin resistant. This is another selectable marker, which is tetracycline resistant. But several other areas are identified, which are the restriction sites, such as ECO-ROI, which is a restriction endonuclease identified from E. coli. This may be cell 1, identified from Saccharomyces lupus. This is another. BAM2, identified as a restriction endonuclease from Bacillus amyloliquefaciens. So in every such type of plasmid, we are getting origin of replication as a very important site. selectable markers such as the antibiotic resistant sites and the restriction sites supposed to be associated with the endonucleases for cutting desired segments of DNA. So this is the design of a typical plasmid recommended as a cloning vector for different styles of biotechnology. Several applicational areas of biotechnology have been apprehended. One of the areas is SCP, which states the full form as single cell protein. Isolated as a very important source of protein rich in amino acids such as D-lysine, D-arginine, D-glycine, etc. And most of the common sources can be dialbicans, say yeast, and one of the very important sources identified as a microbe named Methylophilus methylotrophus. The name is very unique. And this bacteria is supposed to be a rich source of very common amino acids such as D-alanine and D-lysine. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.